This is a Component 1 Taskbook, and we are up to question 4, Photosynthesis. And here is a photosystem. If you've done plenty of Moodle questions, you will recognise this, and we'll have no problem. So A and B, I'm going to say this is our light harvesting complex. So light harvesting complex, and a few different names for that, containing our accessory pigments and so on. You see some different sort of shades there. And B, that's our reaction centre containing chlorophyll A. So reaction centre chlorophyll A. So either of those two do there. Two pigments you'd expect to find in region A. So our accessory pigments, things like, carotene, and you'll have done these with your chromatography and identified these. The special one with the X, xanthophyll, what a great name. But also you can mention chlorophylls that you might find in there, chlorophyll A and B. But these are typically your accessory pigments. And where would you find photosystems? And it says exactly where. So if we've got a cell with a cell wall. In there we might have a nucleus, and here's a chloroplast, double membrane chloroplast, and in there we've got thylakoids in there. So we'd expect to find this on the thylakoids. Thylakoid membranes. Okay, inside the chloroplast. So we've mentioned chloroplast and exactly where in the chloroplast. So two marks there, and these are the two bits. You identify the organelle and which part of the organelle. And then they're shining a light at a plant inside a black box, and they're measuring oxygen of this this plant in a test tube full of some sort of solution. And they're connected to a computer, and the computer will detect. This is an electrode, so this will generate an electric current, and the electric current will be dependent on the how much oxygen is there, and the computer will time it and see how much is produced over how much time. And we've got a coloured filter there, so you might have seen this with your algae balls experiment with different filters and different wavelengths of light. So explain how the use of dissolved oxygen electrode allows the rate of photosynthesis. The key is rate here, and it's sometimes difficult to explain rates. So what's oxygen got to do with the rate? Well, you might have counted bubbles in an experiment, so it's just another way of counting bubbles really, but using this electrode. So why do we use oxygen? Because oxygen is a waste product or produced in photosynthesis. So oxygen is produced in photosynthesis, and the rate of oxygen production will be proportional to the rate of photosynthesis. So rate of O2 production is proportional to the rate of photosynthesis. So more more oxygen, more photosynthesis, that sort of idea. And quite a few people have identified the kind of oxygen is produced, but not really related it to the rate. Two sources of inaccuracy with this experiment. This was pretty hard. You kind of got to think of your controlled variables in any experiment, and you can look at this and go, right, what have they controlled, what have they not controlled that might affect the rate of photosynthesis. So things that affect the rates of kind of reactions, things like temperature, substrates, anything else that might affect it. But so temperature is a good start. We know the rate of photosynthesis will be affected by temperature. I can't see any way of controlling this here. In the lab, you're always using water baths for this, that, and the other. So one inaccuracy could be temperature is not controlled. Correction, use a thermostatic water bath. So thermostatic keeps the temperature the same, water bath. Other things that might affect the rate of photosynthesis, well, you've seen Miss Knight's video on factors that affect the rate of photosynthesis, and in there was, the substrate CO2. Presumably this plant is using up CO2, and there's no way of replacing it. There's no bubbling of air through this, so the rate of reaction would presumably decline over time as the CO2 is used up. So CO2 is being used up, and the correction could be add CO2, some way of adding CO2 or adding a chemical that will produce CO2 or that sort of thing. Add a CO2 source. Other possibilities, probably a bit more tenuous, but the filter might not be exactly right for the wavelength, because it's just a coloured filter rather than a complicated light source that produces a specific wavelength. But go for the two obvious ones there. Draw the results you would expect to obtain for this experiment using a series of coloured filters on the graph below. So this, let's do the easy thing first. Wavelength against the rate. I know the name of two graphs, both with the name spectrum. And which one is it? I know absorption spectrum and action spectrum. The rate is the action. I always think of, action is to do with the rate of things. That kind of makes sense. And so action, and what it wants is you to sort of sketch. My goodness, sketch a graph on here. So a big sort of peak sort of there, and then sort of, I don't know, something like that, corresponding to this is our blue part of the spectrum. This is kind of green. Probably go down further. Actually, probably go down further there, and that sort of, that would be better. If you drew this in pencil rather than pen, then you can rub it out and go for it. And we've got kind of red areas there, and slightly bigger in the blue than the red. That's sort of what it wants there. For 13 marks, a tricky graph, but you have seen it. Even I can't draw it right most of the time. Tricky little experiment, but in any experiment you're looking for controlled variables and just a bit of theory there, so not too bad.